Good afternoon, everyone, and welcome to our first meeting of the new year of the Autism Consortium. My name is Kate Thompson. I'm the Senior Administrative Program Coordinator of the Autism Consortium of Texas LEND program at the Texas Center for Disability Studies at UT. We are very excited today to have Dr. Jenny Hsieh presenting on stem cell approaches to understanding acquired and genetic epilepsies. Before she gets started, I just want to let you know about our next meeting that's going to be coming up on Friday, February 10th at 12 o'clock from 12 to 1:30 on Zoom. We're going to have Dr. Leslie Neely, Hannah McNall, and Melissa Svoboda. I'm so sorry if I mispronounced the name, Svoboda, presenting on the autism landscape in San Antonio. So more information will be sent out about that soon. At the end of today's presentation, an evaluation link will be in the chat. Please take a moment to click on that and just fill that out. It only takes a minute before you close out today. So I am very pleased to introduce Dr. Jenny Hsieh. She's the founding chair of the Department of Neuroscience, Developmental and Regenerative Biology in the College of Sciences at the University of Texas at San Antonio. She's also the founding director of the UTSA Brain Health Consortium. She holds the Semmes Foundation Distinguished Chair in Cell Biology. She and her trainees studied many of the genes responsible for newly generated neurons in the adult mammalian hippocampus. Her work showed that aberrant neurogenesis contributes to temporal lobe epilepsy. Her most recent work focuses on using human brain organoid models to study neurodevelopmental and neurodegenerative disorders. Among her proudest professional achievements are mentoring young scientists and advocating on behalf of women and underrepresented minorities in science. And so without further ado, I will turn it over to Dr. Hsieh.
All right, well, thank you very much, Kate. I am very pleased, delighted to be here, and thank you for Sandy for the invitation. I met Sandy when we were serving together in the same session at this recent UT Brain Health Summit, and I'm very excited to have an opportunity to tell you all about some of our work today. I'm from UT San Antonio, not that far away, and what we've been really doing is trying to use different stem cell approaches, both in vitro and in vivo, to model acquired and genetic epilepsies, as Kate alluded to. I really want to focus my attention today on some of our recent work using induced pluripotent stem cells, which I'll describe in a minute. And these really kind of new types of cell cultures that we can grow these stem cells in 3D, which kind of more mimic how brain tissue is developed naturally, in order to learn about how specific genes contribute to cellular processes and brain development, specifically aspects of human brain development. The other part of my talk also uses mouse models, specifically looking at neural stem cells in the adult hippocampus. We use that to model temporal lobe epilepsy, but really for today, I really decided to focus more on these brain organoid models, and I think they're going to be very powerful tools for modeling a variety of neurodevelopmental disorders, epilepsy, but also including autism. So first of all, I think most of you are familiar about how common epilepsy is, both worldwide and also here in the US. While we know a lot about some of the mechanisms that contribute to epilepsy, and we have a list of good anti-seizure medications, there's still a number of patients that are sort of don't respond to some of the current available medications. So there's still the need to understand some of the causes of epilepsy, particularly during development of the brain, and also understand the genes that contribute to this. So hold that thought. So I also understand, and again, this is very exciting for me to be able to speak to this group, is that there's a common co-occurrence of epilepsy and autism spectrum disorder. In fact, it's been estimated that about 50% of individuals with ASD also have epilepsy, and epilepsy could sort of these spontaneous unprovoked seizures could both precede and follow the occurrence of autism spectrum disorder symptoms. So one hypothesis for this common co-occurrence is perhaps that there might be a common denominator, a common neurobiological basis for these types of disorders. And there's good evidence supporting this common mechanism. For example, in some genetic conditions such as Fragile X or like tuberous sclerosis, other syndromes where single genes, like for example, the FMR1 gene in Fragile X, or genes in the mTOR pathway in the case of tuberous sclerosis, MECP2 in the case of Rett syndrome, so on and so forth. In those specific conditions, there's frequent co-occurrence of autism symptoms and epilepsy symptoms. And so in these cases, we can begin to dissect how those specific genes may contribute to changes in development and the circuits by which those neurons are involved in, and how that contributes to some of these symptoms that underlie, both seizure occurrence, seizure development, and also some of these other types of changes in like comorbidities, cognitive changes, changes in emotion and mood, etc. And one of the thoughts is that perhaps these common neurodevelopmental deficits are due to changes in the inhibitory circuits, or even an imbalance of excitatory and inhibitory circuits. And one gene that is thought to contribute to this is the ARX gene. Okay, so again, my lab has, we've decided to focus on one gene, and it turns out we are focusing on ARX. And the reason why is because, well, first of all, it was, there's many genes that have been identified, particularly through next generation sequencing, that has been associated with childhood epilepsy. And this is just sort of an outdated list of some of those genes. If I haven't had the opportunity to overlay these genes with some of the genes that have been frequently found in autism, but I would suspect that many of them do overlap. And so again, we decided a couple of years ago to focus on this one gene, ARX. ARX stands for Aristillus-related homeobox. It is a transcription factor, which means that it binds DNA and it regulates other genes. And in fact, the gene ARX has both, it really controls both activation and repression of many other genes. And so that mutations in this gene ARX, some of the deficits could be due to its direct binding genes, but it also could be due to other genes that it regulates, which are also other transcription factors. And so if you look at the structure of the gene the protein that's encoded by the gene ARX, there's, in addition to the part that binds DNA, this homeodomain, there's also these polyalanine tracks, or PA1, PA2, PA3, and PA4. And it's, in fact, mutations in the polyalanine tract 1 and 2, shown here, are linked to epilepsy. And depending on where the mutation is, there could be more severe or less severe or mild forms of the seizure disorder, or even intellectual disability. And there are, so the question is what do the polyalanines do, right? So there's, in fact, in the typical or in the normal situation, there is a repeat of polyalanine, but in the case of that's been linked to, for example, epilepsy or even autism, the numbers of alanine is expanded so that it can even go up to like 20 alanines in the tract 1 or tract 2, but it never seems to grow or expand more than this. So unlike polyglutamine disorders that we've seen in like Huntington, where we may get even hundreds of copies of the glutamine, these polyalanine expansion is more limited. affected. And they do tend to fall into these transcription factors, and many of them are associated with developmental disorders. So in and of itself, it's sort of a fascinating gene to study. And this gene is found on chromosome X, so that the X chromosome, so that all of the patients that have been described to have polyalanine expansion and have epilepsy or intellectual disability are males. Okay, so then we have learned a good deal about the role of ARX and which cell types it affects from mouse models. In fact, some investigators have attempted to model the human, the patient mutation, this expansion of polyglutamine by knocking it into the endogenous ARX locus of a mouse. And what scientists have found is that this is a marker here shown on the right of a GABAergic interneuron marker, and these inhibitory cells are found to be less in this expanded polyalanine mutant mouse. So it suggests that perhaps the development or the migration of these GABAergic, these inhibitory interneurons, are affected from the expansion of this polyalanine. And so just to remind everyone, in mouse development, similar to human brain development, the inhibitory interneurons are, the progenitor cells are found in the ganglionic eminence, the lateral and the medial ganglionic eminence, and they migrate into the cortex early in development. So the various interneuron cell types, like the parvalbumin or the calretinin, et cetera, et cetera. And so it's thought that the role of ARX, or the ARX, functions importantly in this developmental process and this migratory process through its regulation of both by activation or repressing other genes, and downstream of this other very important set of transcription factors, the DLX genes. Okay, and these polyalanine expanded mice that have been generated also develop seizures, so reminiscent of the patient phenotype. And so, one possibility is to use these mouse models to begin to understand and also to develop, therapeutic strategies to target these types of epilepsies. But then again, is this disorder specific, or the seizure specifically due to a defect in GABAergic interneurons? So another group published that if you just remove the gene from the cortex using this conditional knockout approach, right? So ARX is found in other cell types, but just specifically remove it from the forebrain using this EMX1 Cree line, which basically conditionally removes it from a particular, part of the brain, these excitatory neurons. It's been shown that there are also changes, particularly in these upper layer neurons that express the markers SATB2 or CUX1, as shown here in the right side, but no change apparently to these deep layer neuron markers as marked by TBR1 or CTIP2. So it suggests that ARX is also important perhaps in cortical neuron development. And this is an important thing to know, which cell types really require ARX and what cell, because when you think about the circuit development, and if you really want to think about, either gene replacement or other therapeutic strategies, having more information about the importance of specific transcription factors and where and which cell types that it's really needed is really informative. Okay, so then, but the question really is, does the mouse models of ARX and other types of genetic epilepsies, how well does it, really kind of recapitulates accurately how a human brain develops, right? And that has been the question I think many of us in the field have been asking. So, and there are really important differences to consider, between the rodent and the human model, and also other non-human primate models, is this really vastly expanded cortex, both the deep layer and the upper layer neurons. There's also this unique sort of expanded population of neural progenitor cells, particularly in this outer subventricular zone. So the rodent has a subventricular zone, but the human has this sort of the subventricular zone, but also this much more expanded outer subventricular zone containing neural progenitor cells. And they're called these radial glial, or these outer radial glia. Okay, so that's one important difference. That's and those cells are not as prevalent in the rodent models. And so again, if the genes like ARX function in those outer radial glia cells, perhaps we're missing that aspect in the rodent models. Okay, another important difference is, as I alluded to in the mouse, the GABAergic interneuron progenitors come from these ganglionic eminence, and they migrate into the cortex. In the human, that also occurs, but there's also evidence where a local source of GABAergic neurons is generated within the cortex. And so these differences are important to consider, especially when you are trying to also develop a better understanding of what happens during human brain development, which again is very difficult to study in a model, okay, using a variety of models. And so that's where we really we're excited and inspired by the use of perhaps these organoid models, these in vitro models that you can directly generate from pluripotent stem cell lines, or these induced pluripotent stem cell lines that can be reprogrammed from blood samples or from fibroblast samples taken directly from patients with polyalanine expansion of their ARX gene. Okay, so that's what we decided to do, is we recruited both healthy controls and patients, children with ARX polyalanine expansion that have either epilepsy or intellectual disability or autism. And we generated their induced pluripotent stem cell lines. And then I'll tell you about how we can generate these organoid models and what they are and what they're good for. But the eventual goal is that we may be able to use them for, but basically for like precision medicine or drug screening to sort of identify personalized medicine that we can maybe give back to the patients in sort of long-term goals. And so today, for today's talk, I'm going to focus and tell you about how we look at their using single cell RNA sequencing technology, unbiased ways of using them to identify cell types, but also corroborating that with using different protein markers that are indicative of different cell types in these patient-derived organoid models. And then after we kind of understand how the cells develop, we can also try to measure electrical properties directly in these organoid models as sort of a surrogate understanding of how these neurons are functioning. And in a way, it's like a window into perhaps how the patient-specific mutation might be functioning during development, which again is something that we can never really study or access. Okay, and so in our study, what we did was so first of all, one of the things we want to consider is how many patients should we recruit, and what are the best controls for these patients? So these every patient is very different. They're different in their symptoms. They're different in their epilepsy, semiology. They're also very different in their when their seizures occur or how their symptoms manifest, et cetera, et cetera. And so before I came to UT San Antonio, I was at UT Southwestern, and we collaborated with a pediatric neurologist in the Children's Hospital. So our first patient was actually patient 1, and this patient has in addition to the normal 12 alanines, as I alluded to, so everyone has 12 alanines in their tract too, but this patient had 8 additional alanines. And so we strive to identify and recruit at least two other patients also with 8 additional alanines. And so we ended up contacting other clinicians at different sites. And so our healthy controls are also males, and we try to match their age. But again, it's not, it's sometimes we just get what we can get. And because of possible differences in the genetic background, besides these polyalanines, we try to, every time we make these stem cell lines and organoid cultures, because of this heterogeneity, we study at least 2 different cell clones per patient line. And we also try to study at least 3 patients and 3 healthy controls. And so that's sort of our strategy. And that seems to be right now the gold standard in the field. But there's another powerful technology, which we are doing now, which is we can try to directly gene edit these polyalanines to basically replace this additional 8 alanine so that we can kind of convert it back to the normal 12 alanines. And this would be an isogenic control within the patient line. And this really controls for all other variables. And those isogenic controls would be really great and ideal to have. Okay, so now with these induced pluripotent stem cell lines that we have in the lab, I forgot to mention we established a stem cell core facility here at UTSA. This procedures can take months to perform, and the quality control and to bank these lines. So that was another resource that we decided to establish here, and it's in the University of Texas system. Everybody can have access to it as a fee for service. So it makes this type of work more feasible. And so then Vanessa Nieto Estevez, a postdoc in the lab, working together with Parul Varma, also a very talented postdoc in the lab, what they do is they take these human-induced pluripotent stem cells, and then they're growing them into brain organoids. So if you're wondering what are brain organoids and how do you make them? So our rationale was to think about what are the neurons or the neuronal cell types that we want to create, that we think that the ARX expansion mutation would affect the most. And so as I alluded to from the mouse studies, there seems to be changes in both the GABAergic interneurons, but there's also changes in the cortical neurons. So sort of based on that rationale, we take these pluripotent stem cell lines and we use factors that will promote neural fate, because we don't want mesodermal or endodermal fate. We specifically want neural fate. So that's the first step, is to subject them to this neural media. But then even when you direct them to neural fate, they can still, become any of a number of different neural cell types, right? Including neurons and glia. And then even within neurons, the decision tree is to subject them, to bias them really specifically toward cortical neurons and ganglionic and GABAergic neurons. So that's where another decision point comes in, where we want to promote kind of more dorsal, which is more cortical fates, or ventral, which is more of these GABAergic interneuron fates. And so using factors that basically bias toward cortical or ventral, these neurons over time will adopt those cell fates, okay? And then, so that's where the neural induction happens, then the patterning and maturation. And then with time, just like, for example, how a brain will develop in vivo, we see, early progenitor cells, and then we eventually see neurons, and then we see astrocytes or other types of glia. And then with longer and longer times in culture, we may see sort of formation of circuit activity. And that's what Vanessa and Parul do. So they take different time points, like 30 days, which are sort of more, they're just again, recapitulating first trimester development, so 30 and 60 days. But then we also take a time point like around 120 days in vitro, sort of capturing sort of that second trimester where you have cortical neurons or GABAergic interneurons. Now, we sort of did our analysis and we stopped it at about 6 months, just because for other sort of logistical reasons that we, because again, you have to balance with how long you grow these in vitro and how long do you, you want to be able to complete some of your analysis and just to see what is happening. But there are other investigators that have reported taking these brain organoids as long as 1 year or even 3 years out in vitro to really get some of that postnatal brain development formation. So it's again, it's very fascinating. There's a lot of changes at various different time points. Okay. So then what we're doing in this first part of the study is we're comparing expression of different genes using immunostaining, but also using the single cell RNA sequencing, which really captures this transcriptome of the cells in an unbiased manner. And so that's what I'm showing here. So this is single cell RNA sequencing. And so what that is you on the left, well, first of all, I'm showing you just cross section of these cortical organoids from the healthy, like an example of a healthy control patient or healthy control. And on the left are sort of cross sections of immunostained brain organoids at 30 days or 120 days in vitro. And the different markers are marking different cell identities. So PAX6 is a marker that is typically found in a neural progenitor cell. And I'm showing you a sort of a zoomed in image here on the left, where hopefully you can see like a ventricular zone-like or a tubular zone-like structure, where there's sort of a hole in the middle and a ring of cells around the outside. And these ring of cells, they're nuclei and they all express PAX6. So, and you can find different, amounts of these little sort of circular structures. So in real developing embryo, you would have a single neural tube, whereas in an organoid, we find a variety of a number of these circular or these tubular-like regions. But it's really fascinating to see that we do see that in these early cortical organoids. And then by 120 days in vitro, as the organoids are expanding and these neural progenitors are differentiating into some of these cortical neurons that express like this marker, CTIP2 or even SATB2, you start to lose those rings or those tubular regions, and they become more uniform. And then again, how does the single cell RNA sequencing match up? So what we do here is we would take that entire, three-dimensional organoid, we would dissociate them into individual cells, and then we would capture each cell into one of these gel emulsion beads. And then the sequencing machine would just basically sequence the mRNA transcripts from individual cells. So in this way, you could then see based on the genes that the cells express to identify the cells themselves. So it's not as biased as where we do it on the left with immunostaining, where you're sort of using different markers. So again, on the right, there's 30-day and there's 120-day, and all the different colors are marking the different identities of the cells. Like pink is a bunch of genes that give you a signature, and the signature defines the radial glia cell, which is a type of neural progenitor cells. And by 120 days, the sort of this dark blue color is having a molecular signature that defines an outer radial glia cell. So it's interesting that we, as we would predict, and what has been observed in human fetal brain development, is that with with time in development, there is an emergence of this outer radial glia cell. And again, we capture this in our organoid culture. And again, these cells are not as prevalent in the rodent model. So it's really exciting that we can detect this from our human pluripotent stem cell derived organoids. And then again, going back to the left, the marker FAM107A and HOPX are, again, proteins that are expressed in these outer radial glial cells. So all of this is to say that using both the single cell RNA sequencing and the immunostaining, we really have validated this cortical organoid culture model. And then same with the ganglionic eminence organoids. So as I mentioned, we can take factors, we can direct them to these ventral identities. These are where the GABAergic neurons originate. And so using markers that are expressed in GABAergic progenitor cells like NKX 2.1, we can find expression of this protein in these ganglionic eminence organoids. Same with the single cell RNA sequencing. We also can identify using molecular, like a set of genes, a molecular signature that defines immature interneurons, and also at 120 days, even a population that expresses somatostatin. Okay, and they also express GABA and other genes that are found in GABAergic cells like CXCR4. Okay, so with these cultured organoids in hand, these models in vitro, we can now ask these questions about what these ARX mutations. So one of the things is we want to also know which cell types in the cortical organoid and the ganglionic eminence organoid, these GEO organoid, that actually express ARX, our gene of interest. So in fact, in the cortical organoids, we find about 10% of those cell types in the organoid expresses the gene ARX. And among those ARX expressing cells, we can look at other genes that are, that define those cell types. And many of them are these outer radial glia cells, these neuroprogenitor cells, and to a smaller extent, they're cortical neurons. And in the GEO organoids, ARX is expressed even more frequently. About 20% of those cells express ARX. And those ARX expressing cells are also expressing interneuron progenitors, these and characterized by these, DLX genes and also LHX genes, and even to some extent interneurons like GAD, expressing GAD1, GAD2, CXCR4, and somatostatin and calbindin. Okay, so again, that confirms that these organoid models we're using contains ARX positive cells. So now we can really compare it to the patient that has ARX polyalanine mutations to determine how the patient mutations affect ARX expression. So in the cortical organoids, we actually find an increase of ARX expression from the patient-derived organoids compared to control, and this is seen using immunohistochemistry. We can also see this in the single-cell RNA sequencing. In the GEO organoids, these ganglionic eminence organoids, we find an opposite effect. So we see a reduction of ARX, both at 1 month and at 2 months. So again, it's interesting that depending on the cell type, depending on the context, cortical versus ganglionic eminence, the level of ARX is either increased or decreased when you're comparing the patient-derived organoids compared to control. So then what's the effect of having these changes of ARX expression? So now we can take a look at the different cell types, right? So we can use markers for proliferation or progenitors or even the different neuronal cell types. So in the cortical organoids, there's actually a slight increase in proliferation. But so we thought, well, maybe there's changes in the progenitor cells. So when we looked at the progenitor cell markers like PAX6, we find a reduction, interestingly, in the patient-derived cortical organoids. So we wondered if this is a reflection of a change in the radial glial cells. And so those radial glial cells express a diversity of different genes. And so we surveyed 2 of them, the HES1 and HES5. And we actually performed RNA scope, which is a way of looking at messenger RNA transcripts directly within the organoid tissue. And we find that there's both a decrease and an increase in this using these markers. And so when we looked at single cell RNA sequencing of this population, in fact, we find that the radioglial cell population is increased in our patient-derived organoids. And if you looked at individual genes, some are increasing and some are decreasing, but as an entire population, this population is expanded or increased. And so I just want to summarize this part of what I have so far in terms of the cortical organoids, and so that we think the ARX polyalanine expansion at this very early time point when there are a lot of progenitor cells that are developing, that having this expansion, depending on the target genes, whether it's DLK1 or HES1 or HES5, there are both, some are increased and some are decreased. But in general, this is leading to like a premature differentiation. So when you look at the next step, which is the cells are becoming neurons, we see some of the neuronal genes, like beta-3 tubulin is a gene that's expressed early during neuronal differentiation. That seems to be increased in our patient-derived organoids. And we think that what potentially might happen if this persists over time is this might actually lead to a sort of a depletion of the stem cells, because they're sort of coming out of the gate, so to speak, a little bit earlier than they should. So to look at that, we decided to look at the next stage of development, which is sort of 120 days in vitro, like a few more months down the road. And sure enough, when you look at this marker of outer radioglia cells, that's decreased in our patient-derived organoid, here as we quantify it. And then when you also look at markers of neuronal differentiation, like NeuroD1, that's also decreased in our patient-derived organoids. And then subsequently, when you look at markers that identify like some of these deep layer or upper layer cortical markers, we also see a trend toward reduction of these cortical neurons. So it's sort of consistent with our hypothesis that at this early time point, when you have this increase in the progenitor cells, they're maybe prematurely differentiating. Now, at a later stage during development, you may perhaps depleted your progenitor cells, and this ultimately leads to a decrease in your cortical neurons. All right.
Can I ask a quick question? I'm just wondering, so when you have these kind of results, what would be the impact on a patient that you're seeing? What would be the difference that a family might notice?
Yeah, so at this point, if I was really going to extrapolate, I would say you're somewhere in second trimester, because we're still looking at very early stages, even with these in vitro models. And at this point, there's not a lot of glia yet. Glia sort of start to differentiate around 120, 150 days. And so if I was really, again, going to extrapolate this out, I would say, well, perhaps there could be a compensatory change later during development. If there's less excitatory neurons, maybe we would have hyper or overexcitation. But again, what's happening with those inhibitory cells, right? That need to respond to this. And from what we know of other models, we know that the ratio of inhibitory cells closely is tuned to the number or the ratio of excitatory cells, right? So this, again, is very reductionistic. It's just looking at the excitatory cells at this point. Okay, so I think let's hold that thought. So let's look at the inhibitory cells, right? The other side of the equation. So we made those organoids, we made these GABAergic, these geo-organoids, and we see a reduction in neurons as evidenced by this marker beta-3 tubulin or TUJ1. We also see a slight trend toward reduction of GABA, this inhibitory neurotransmitter. And interestingly, we see also a change in this CXCR4, which is also a marker of interneurons. So wanting to, again, I sort of alluded to, we were at this point, we're just looking at the cortical and the geoorganoids separately, but in the brain, they're not, these cell types are not developing separately. They're interacting, the interneurons are migrating into the cortex. So here we actually have this very exciting opportunity to model cell migration in vitro, so we can grow the cortical and the geoorganoids separately in wells, but we can take a virus that will express this interneuron reporter gene that's fused to a green fluorescent protein, so that we can watch the migration of these labeled interneurons from the ganglionic eminence side into the cortical side. So trying to model that very early interneuron migration that we would typically never be able to see in an intact human brain. So this is what we do. We actually position these fused organoids, people call them assembloids, directly on top of a microscope objective, and we can take images of these fused organoids over time. So in like a 18-hour recording, we will take an image every 20 minutes. And so if you can, these are just static images of the labeled interneurons fused to this green fluorescence protein, or GFP. Before fusion, all of these interneurons are on the cortical side. And then after migration, this would be 9 days after migration or after fusion of these organoids, these interneurons have moved into the cortical side. Okay. And so here are just, I made some of these movies. This is like an example of an overnight recording. And I'm imaging sort of that part after it's moved in. And I'm only showing you a video of an overnight recording, maybe about 18 hours. So it's really sped up. Right. So in real time, you really, these interneurons are moving much slower than we can see by our eye. But if you take images, every 20 minutes and you speed it all up, you actually can see that they do move quite a bit. Some of them become blurry because they're outside the field of the plane of imaging. So what we can do is we can now measure the distance and the speed by which these interneurons are moving. And what we found is that in the patient-derived organoids, they're moving faster and further. So that's an interesting and also kind of different than what people have reported for ARX mutation in a mouse, where there is lack of interneuron migration. So we wanted to see, well, is this putting all of these averages of the distance together, the ARX, the control is labeled in green and the ARX is labeled in yellow, and the ARX patients those neurons are moving faster and further. So again, is this due to the mutation in the interneuron organoids themselves? So we can do these mix and matching experiments. So in the version where we just make an assembloid where the ARX mutation is in the interneurons, and we have them move into just healthy control cortical organoids, we find that this is labeled in red, and they are also moving at a faster and further, similar to the ARX fusions. And then finally, we make a fusion where just a control interneuron organoid is moving into an ARX cortical organoid, and those are labeled in blue, and those are very similar to just the control fusions. So this really reinforces that the accelerated interneuron migration is due to the mutation being in the ganglionic eminence organoids. So then putting all of this together, what does this have? I think the question of what does this mean for the patient? Well, again, we don't really know what this means for those individual patients, but in the patient-derived organoids, we can begin to measure neuronal activity. of these organoids. So we basically take those assembloids and we put them on these multi-electrode arrays. And so at the bottom of these wells, there's 12 electrodes that are measuring extracellular neural activity. And so if we just look at the electrodes that is contacting these organoid neurons, if you look at all of the population events, so those neurons, they're spiking and bursting, and there's not an obvious difference in the number of events or the amplitude of those spikes when you look at the ARX control versus the control. But if you actually look at the power frequency of those events, and especially if you look at the most active organoids, if anything, the ARX organoids are less active, suggesting that the polyalanine expansion in these interneurons is decreasing their overall network activity compared to the control organoids. And so then again, I think just trying to put this into some kind of context, earlier I showed you that there's changes in the cortical organoid, there's less cortical neuron developed, but in this ganglionic eminence organoid, there's also less GABAergic interneurons developing, but there's also a change in their migration so that they migrate faster and further into the cortical organoid. And so And why are they doing that? Are they doing that because they're not finding their partners, their synaptic targets? Is that why they keep moving or migrating? And if less connectivity is perhaps what might be explaining this overall reduction in neuronal activity, that's a possibility. And then how does that relate to epilepsy when we think of epilepsy as too much synchronous activity, right? So, and so again, this is just a window into, like a really short overnight recording. So it's possible that maybe more extended recording. And again, we chose this very early time point of development. Maybe the outlook would be very different later on in development. So we can definitely talk about that at the end, but I do want to make sure I have time for questions. And I also want to frame this in a bigger context. So as I alluded to, here we have an opportunity to go directly from patients and be able to model perhaps their brain development and maybe even their circuit function in a dish. So can we use these models to improve treatment outcomes? So that's the question I'm asking here. So here is some examples of anti-seizure medications, that have been discovered and that are currently under, clinical use. And a lot of the use of these anti-seizure medications are based empirically, where there's a clinical diagnosis, and there may or may not be genetic assessment or gene panel testing, but a lot of that information is not really used as well to design or tailor their anti-seizure medication. So the idea with the precision medicine approach is based on both the clinical diagnosis and the underlying genetic data that could be used to guide potentially the use of, the tailoring of the medication, or even the design of screening of certain medication. But that would really need to involve in vitro and in vivo functional assessment of these different genes or even variants as they are being, uncovered. And then using that information to sort of guide or dictate even, drug development, and those types of approaches to then go into clinical trials and a sort of a more precision-based treatment. And so I think moving forward, the idea is that, this is sort of the empiric treatment that we have at hand, with every medication that is prescribed, the efficacy is decreased, so that, you know, there's hundreds of thousands of patients that are medically refractory, but that if we had, you know, these types of in vitro models that match the patients, these individual patients, and we can use them to assess the functionality of these genetic variants, you know, using these different readouts, cell-based readouts or electrically based readouts, that perhaps, you know, one day the hope is that this could lead to sort of more precise medication, or even with even the current available medication, or even combinations of drugs, and possibly even screening of new drugs. So, but still, there's still a lot of limitations to this approach. I've already kind of alluded to this. You know, in our recordings at this point, we find less activity, and that's, you know, how does that translate to epilepsy? Again, we are really studying a very early stage of this process. Our organoids are still very immature. They don't contain a lot of the interneurons that you would typically see as the brain further develops and matures. And also, we are only at this point looking at two different, you know, brain regions, so to speak, you know, ones that resemble cortex and one that resemble the ganglionic eminence. There's other regions of the brain, and certainly as stem cell biology is also progressing in terms of the different protocols to make other regions that and to form other circuits, you can imagine connecting other regions of the brain to like for example, hippocampus would be one obvious target to kind of better recapitulate this complexity. And so other organoid models in conjunction would be very important, and also the goal of the future. Okay, so with that, I just want to acknowledge the people in my lab who did the work. I mentioned Vanessa and Parul, but also, you know, our team of electrophysiologists, and then we collaborate extensively with our clinicians at UT Southwestern, at Children's Hospital, Eric Marsh, and Jeff Golden at Cedars-Sinai for the ARX project, and also our stem cell core. And also, in addition to our stem cell core, we also use extensively our genomics core for the single cell RNA sequencing, and also the UT Austin sequencing core. And I also want to acknowledge the patients and their families for really being supportive and amenable to this type of work, without which we can't do any of this is possible, and also my financial support. So with that, I'm really happy to just talk about any of your questions, and to have a better, more discussion about this topic. Thank you very much.
Thank you, Dr. Hsieh. If anyone has any questions, please feel free. You can either write it into the Q&A function or into the chat, or if you would prefer to have your be unmuted and speak out loud, we welcome that as well. Just let me know in the chat. I do see somebody has a hand up. Let me find who they are, or at least did. I don't see it now. If somebody had a hand up a moment ago, you could raise that again, or just, oh, Angela McCarty. Okay, I'm sorry about that. Let me see how to talk. Okay, Angela, you can go ahead.
Yes, hi, Dr. Hsieh. Thank you for that insight. I had a question about one of the slides where you referenced like the stem cells. Like, do y'all use human stem cells to do certain research? I know there's ethical things too when it comes down to stem cells and things like that. So how do you use stem cells? In what way?
Yes, thank you for asking this question. I don't know if this is the slide you're talking about, or it could be this slide, but yes, these are human-induced pluripotent stem cells. And, you know, I think I really should have mentioned this more, but you're absolutely correct that that pluripotent stem cells, which are the cell types that can ultimately differentiate into all of the hundreds of cell types in the body, including our brain cells, they normally coming from the embryo, and particularly this blastocyst stage. And there are certainly some ethical concerns, right, related to using these embryonic stem cells. But what I forgot to mention is that in starting 2006, that scientists have identified the technology. It's specifically for transcription factors. It's like a cocktail that you can convert any cell type, including blood cells, into these pluripotent stem cells. And so these are sort of artificial, right? They're induced because, they're done outside the body. And so these induced pluripotent stem cells don't require the use of embryos. So it really does bypass, I think, some of these ethical concerns and allow us like a window into development that otherwise would be very difficult to study.
Okay. Is that device, some stuff I know a little bit, CRISPR, is that the CRISPR technology y'all use for that, or is it a different kind?
It's a different kind, but CRISPR, is also, as you've probably heard, is a very powerful gene editing tool. And it allows us to very quickly scan the genome, find the mutation, and edit like a word processing document that change into some other back to sort of the normal sequence. And that technology works more efficiently when the cells are dividing, and stem cells are very amenable to gene editing because these induced pluripotent stem cells divide very rapidly. And so we are combining CRISPR in these induced pluripotent stem cells to be able to test how editing the particular mutation might, rescue that particular phenotype to really see if that change has any pathological significance or not.
Oh, that's great. And then you mentioned also some in your lecture about precision medicine. Can you elaborate a little bit more on how that works for this type of research?
Absolutely. So I would say that the term precision medicine, I think, came from the cancer field where the identification, like, for example, in different types of cancer, based on the genetic change that caused or contribute to the tumor, different drugs specifically target those genes. Right. And so and that has been very powerful for cancer treatment, right? Tailoring the drugs to match the effect of those drugs on the specific genetic mutation that the patient has. Now, for autism and even for epilepsy, that's really like our hope, right? To be able to be, based on, I think, the success of the cancer field, see if we could also do that here for neurodevelopmental disorders. And so for ARX mutations, I will say that there are no precision medicine yet that have been developed that specifically treat these ARX mutations. There are some sodium channel mutations that are, I think, and potassium channel mutations are, I think the ion channel mutations have specific drugs that already are being used. And so now people are matching them to see which ones might be more efficacious depending on those particular ion channel mutations. But I really, at this point, I think it's still very much under investigation.
Thank you very much. Thank you.
If anybody else has a question and would like to speak, please drop that in the chat. Let me know. This is all just such interesting research. About how long on this particular study, about how long of a period of time has this been conducted over?
So what I talked about today, I think we started in 2017 and we've finished the manuscript, and right now it's under review. And everything I'm showing you now is what's in the first paper. But the next step is I really want to go from the organoid model to the drug screening. And I think that we need a better, more reliable tests of function or the physiological properties. And so optimistically, I would say another 3 to 5 years. But I think the other thing is this is only track 2 mutations we're studying, and many of the patients are also have expanded track 1. And I've had patients contact me, and they've also contacted Jeff Golden, our collaborator at Cedars. And so we would like to renew our grant, first of all, and second of all, then be able to study patient mutations in track 1 to see how general or what broad some of the things we're finding is. I think Sandy has a question. Hi,
Yes.
Sandy.
Oh, hi, Dr. Hsieh. This is Sandy Magana, and I just wanted to say thank you so much for coming and presenting this to us. I think the Autism Consortium, we really want to present, science and research, and as well as other things, programming related to developmental disabilities, and this is really bring us right into the science, and I think it's super important. I didn't have a particular question, but just want to let you know I kind of joined on late because I had another activity right up until noon. But yeah, we really appreciate you coming.
Yes, and as you know, you and I and others are Leslie Neely, Maria Sharour, Melissa Svoboda we're starting to talk together and work together in order to see if any of these models combined with genetic identification of all of the sort of heterogeneous autism subtypes that we might be able to really kind of move the needle in this area. So I'm really excited to be able to work with you all.
Yeah, very exciting collaboration.
Great, thank you. Does anybody else have any questions, any comments? All right.
I think two.
I have one question just came up. It looks like Greta started in the, oh, there we go. There's Greta. Here we go. I'll allow you to speak. Go for it.
Hi. You mentioned that about 50% of people with autism have epilepsy that either shows up before or after the expression of autism. But, a lot of times before would be when the person is like, less than 2 years old.
Mm-hmm.
If a child has not been diagnosed with epilepsy, I mean, what sorts of signs that epilepsy could have been present before the autism diagnosis? I think that a lot of times what parents face is a lot of question marks about what's going on with their kid, and there's also sort of a constant looking back at what happened here, sort of a post-mortem, ongoing, and so I just wonder, like in terms of like, say all of the diagnostic questionnaires that are asked to parents and how we could better design those, given, because I didn't know that. And actually, I have some sort of other observations about this link between autism and epilepsy. And a lot of times it's, So anyway, that's my question. Thank you.
I think that's from what I've read in the literature, that depending on the study, it's about when if any of the patients that have autism or have the autism diagnosis experience seizures before their autism diagnosis. And so again, I don't really, I'm not sure I have because I do more basic research and I wasn't involved in those diagnostic studies. I'm really not sure how those questionnaires were designed or if they were sort of more retrospective studies.
Mm-hmm.
And I don't know, perhaps maybe somebody else in this group maybe could address that. But I think it's a really important consideration, just the sort of how, sort of the etiology and
Yeah, because I bet that sometimes these seizures go unrecognized or, you know, yeah, because parents don't know what they're looking at. They just,
right.
Yeah. So anyway, well, thank you so much. This is really interesting.
Thank you, Greta. Does anybody else have any questions or any comments? Let's see, I know, Sandy, I see your hand up still. I didn't know if you had another question. Did you have another question?
I didn't have another question, but I forgot to lower my hand. But
Oh,
I did want
okay.
to say that in response to Greta, yeah, I think that, you know, usually it's the neurologist or pediatric neurologist that will diagnose, you know, seizures or epilepsy. But sometimes, I mean, one of the challenges is pediatricians don't always know everything if that's the first point of contact that you're making, and often don't even know about autism. And many children don't get diagnosed till they're 5 or 6, till they're in school. So so there's plenty of time for for other things to be diagnosed or developed. But yeah, I don't know if there's some sort of, there's a lot of different questionnaires and observation things that are used for autism that don't necessarily ask questions about seizures or epilepsy.
That is very true. And it's definitely important information, I think, for all of us to have. I don't think it gets nearly enough attention, you know, especially, you know, among families in the autism community. Just had one comment in the Q&A, just thanking Dr. Hsieh for her time and for her incredible work. And let's see if anybody else has a question or a comment. I see Greta's hand up again. Hang on 1 second. Greta, did you have another question?
Oh, it's just maybe more of a comment. I just wanted to mention that in the case of my son, we had ruled out epilepsy year after year after year, ruled out seizures year after year after year. And now as an adult, we find that that is the case. It's not called epilepsy, but and even so, the seizures are not easily identified on any sort of diagnostic tool that they have. You know, it's not like, you know, they show up to take a picture at the time that anybody wants to you know, so yeah. So I guess I just wondered about that early those early diagnostic tools and how to improve them, or, you know, so that, because families often, they just don't know how to answer those questions. Even the questions are pretty vague, you know. Yeah, do they ever make eye contact? And if they've made eye contact once, then that's not, you know, it's not really a, you know, doesn't fit the criteria. So, but meanwhile, you know, lots of kids go undiagnosed or underdiagnosed, or, you know, get to be 15 years old and then there's a diagnosis. But thank you again.
Thank you for your comments. Okay. Well, I just want to reiterate, thank you so much, Dr. Hsieh, for coming and presenting with us today. I do have one more question in the Q&A. is wondering how someone might get their granddaughter diagnosed or tested.
Is this
Is that
question
something that you were for you? Yeah, what is that something that you know, would go through someplace like your lab, or if somebody was interested in having this, you know, any kind of this testing done, what kind of route would they take?
So our we collaborate with like Eric Marsh at the Children's Hospital of Philadelphia, and also Jeff Golden, who's at Cedars. And I think depending on, you know, sort of like what Sandy was saying, depending on your pediatric neurologist, if that's probably the first step is to talk to them and then see if you know your granddaughter is a candidate for genetic testing, either genetic sequencing or a gene panel. So many of these, either if it's for autism genetics or epilepsy genetics, but a lot of the genes overlap. And so you can inquire about that. So I know the ARX is on these panels. So that's probably what I would suggest, but I'm sorry, I'm not a clinician, so I'm not really, yeah, I don't feel that I have the expertise in that department.
Yeah, thank you. Well, if I don't see any more I got one more hand up. Okay, there's Sandy. Go ahead.
Yeah, absolutely what Dr. Hsieh said about diagnosis, but in every area, every region, it's going to be different what the resources are, right? And it depends on what you're seeing. If you definitely want to go to your pediatrician and ask them to refer you to pediatric neurologist or a developmental pediatrician. Developmental behavioral pediatricians tend to diagnose autism and are very expert in that, and other developmental disabilities. They may not be as expert in epilepsy. So I would recommend the neurologist for epilepsy and the developmental behavioral pediatricians for autism and developmental disabilities, but your pediatrician may have some skills or knowledge in that area. If they don't, I would definitely ask for referrals. And this is a challenge for many families. They don't know they should ask. And a lot of times they just accept what their pediatrician tells them. It's really important to advocate and push for things that if you really think that, you know, there's something else there.
Absolutely. Yeah. That I think learning about advocacy and how to advocate for yourself, how to advocate for your kids is definitely an important skill set you start learning pretty early on when you have a child diagnosed with autism or another neurodevelopmental disability, and always kind of push for sure.
Yeah, I mean, I think I can imagine how frustrating it is. In fact, I know a scientist in my field, she's a huge patient advocate now. Her name is Tracy Dixon Salazar, and I don't know if she's spoken to this group, but her daughter developed seizures. And she said that her pediatrician, her neurologist, like no one could explain what was causing this. And then she ended up quitting her job, going to get her PhD, sequencing her daughter's genome, identifying this calcium channel mutation. And that led to the diagnosis, but then treatment in terms of, you know, varying the amount of calcium in her daughter's diet. And then she started the Lennox-Gastaut Syndrome Foundation, LGS Foundation, and she's a big patient advocate and of course a scientist. So it's a remarkable story, but she always talks about how incredibly frustrating and challenging it is.
Absolutely. Yeah, that's a route to take that I don't think most parents have available to them. You know, but that I think that's I have heard of her in that work, and I just think it's incredible to be able to find that to be able to kind of just keep pursuing answers like that. Okay, well, thank you again so much for coming and speaking with us today. Everyone else, please remember I will post one more time in the chat. Please remember to go to our evaluation link today. Go ahead and click on that so you have the window. And with that, we will see all y'all next month on February 10th. Right.
Thank you very much.
Bye.
Bye, everyone.
Bye.